The topic of mutation and gene editing brings us to a point in the course where we really get to look at some of the forefronts, or the area that biology is expanding the most, in my opinion. Biotechnology and genetic engineering is really taken off in the last couple of decades because of the development of some new technologies, and the possibilities that these new technologies bring are really astounding, and all kinds of new research and potential possibilities to cure diseases, to change human lives, to change other organisms' lives, are really due to advancements in genetic engineering and gene engineering, or gene editing. And in this video, we'll look at mutation specifically, as well as some of the advancements in gene editing and engineering, and this is for D1.3. So let's first start by addressing what is a mutation. The DNA itself as a molecule is really well protected. Inside of the nucleus, it's protected and is not varied or changed due to external conditions, external factors. Typically, it's very stable within the nucleus, and there's an extremely small rate of mutation that does take place during DNA replication, about 1 in every 100,000 nucleotides. And so a mutation is simply a change in the sequence of the DNA. They are random. Mutations are random. It's not a deliberate change. Organisms cannot decide that they need to have a mutation in order to be able to survive. Mutations are completely random. This is obviously disregarding the human purposeful gene editing that we'll talk a little bit about later. There's a couple different types of mutations, the first being substitution, and this is pretty straightforward. This is when one base is replaced by another due to changes in the bases or mispairing. And the most common example, the most common mutation then would be is changing a guanine to a thymine. And this occurs because of a slight shift in the position of the nucleotide in space or inappropriate bonding. The second type then is insertion, and this is the addition or insertion of a nucleotide so that there's an extra base. And this requires a pretty major change because it shifts all of the nucleotide bases down, so it changes up the sequence. In order for this sort of mutation to take place, it requires the breaking of the backbone of the sugar and the phosphate backbone of the DNA molecule in order to be able to occur. And then the last type is a deletion, and this is the removal of a nucleotide, so essentially the opposite of insertion. It also does require the breaking of the sugar phosphate backbone. In the process of DNA being used to make proteins, the DNA sequence is read and copied into a strand of messenger RNA that then is actually used as the code to be able to produce the protein. So it's kind of a middleman between the DNA and the actual production of a protein. And that messenger RNA is read at 3 nucleotides at a time. It's looked at or read by the ribosome in order to produce that protein, and it reads 3 nucleotides at a time on the messenger RNA, and that is referred to as a codon. And that codon calls for or codes for a specific amino acid. And so a codon is 3 nucleotides on the messenger RNA, and that messenger RNA is a copy of the DNA. And this will be further discussed in D1.2. Within the DNA sequence and the messenger RNA sequence, there are different types of mutations that can have different effects or consequences, both positive and negative. Additionally, not all of the DNA within humans or any species is actually responsible for producing proteins. And within humans, there are single nucleotide polymorphisms, or called SNPs, SNPS, abbreviated SNPs, and these are base substitutions, mutations within the human population at a single base position that's happened at some point in the past, and it occurs in the non-coding regions of DNA, so the sections of DNA that are not responsible for producing proteins directly. They're sometimes associated with certain diseases, and the correlation of the presence of these SNPs and the disease itself allows for the determination of one's predisposition to develop a specific disease. So these can be looked at to draw some conclusions. And so within, whether it's the SNPs, the non-coding sections, or coding sections, mutations can have some different types of effects. And one of those we call same sense. And this is a change in one codon for an amino acid into another codon of the same amino acid. Many codons actually code for or provide the instructions for the same amino acid to be put into the protein. And so a mutation that is the same sense is not going to have an effect on the phenotype. It's still possible for a future mutation to be able to change the amino acid, but at this initial mutation, it does not have an effect. And this distinguishes or highlights the ability of different codons to code for the same amino acid, and that really helps to protect against the impact of random DNA mutations. If you think about a mutation from an A to a G, for example, just a random mutation, then if that doesn't change the amino acid, then it really has no effect on the organism, and this allows that genetic code to be continued. Codons that specify the same amino acid usually differ by just one nucleotide, and amino acids with a similar chemical structure are encoded by similar codons. And not only does the proofreading of DNA replication that we talked about earlier help to reduce changes, so does this actual code help to ensure that the sequence of amino acids that make up that polypeptide chain are conserved. The second type is called a nonsense mutation, and this is a mutation that creates a stop codon. And so codons can have starting or stopping points. All start codons are AUG, which codes for the amino acid methionine. The stop codons, there can be a couple different types, and a nonsense mutation then it creates nonsense. It develops a stop codon, and therefore it stops the translation process, the production of that polypeptide, before it's complete, and it limits or prohibits the function of that protein. The third type is called a missense, and this alters one amino acid within the protein, and the impact of this can depend on the type of alteration and the function of the protein. It can have a minimal effect, or it can have a major effect and be lethal. Oftentimes we see this cause genetic diseases such as sickle cell anemia. If it's conservative, the protein is functional. If it's non-conservative, the protein is non-functional. So depending on where and which amino acid is being changed, sometimes this can cause the protein to continue to work, continue to function, or sometimes not. Very rarely does it actually improve the function of a protein or increase the chances of survival. If the mutation is then inherited by the offspring, the new allele of that one gene is produced and increases slightly the genetic diversity of the population. So this is how new allele types of a particular gene can be produced. And this can actually be, if new genetic diversity is produced, it can be beneficial for the population from an evolutionary standpoint. Insertion and deletion mutations are less likely to have beneficial consequences compared to substitution mutations. They almost always result in the coded protein not functioning because it causes something called a frame shift mutation, in which all of the following nucleotides after that mutation are changed or read differently because nucleotides have been added or deleted. In the image, we can see here a deletion in which the amino acids produced are completely different after the mutation than what would occur in the normal sequence, the non-mutated sequence, because of that deletion. Insertion and deletion of 3 nucleotides, which is possible, are not frame shift mutations, but they still add or remove one or more amino acids from the protein, and then this can cause actual structural changes and functions within the protein. A good example of this we can see in BRCA1 gene. You've maybe heard of this before. It's often associated with breast cancer. It can be referred to as the BRCA1, and this particular gene is a tumor suppressor gene, and those types of genes are used to repair DNA. Specifically, they mend double strand breaks and can correct base pair mismatches. And so if this gene is mutated, it can cause an increased risk of other mutations because that correction function is lost, that ability to repair is lost. And so this can lead to, not always, but can lead to tumor formation, eventually cancer, particularly in the breast, ovarian, or prostate, and it can lead to the cancer in those 3 areas. There's about 20,000 alleles that have been identified, or different types of this particular gene, including a variety of substitution, deletion, and insertion mutations. Some of these have no effect. Others actually do create an increased risk of cancer to varying levels. As previously mentioned, mutations are random. They can occur at any time, and most often we see them take place during DNA replication. That's the situation or the time of greatest risk. External factors or agents called mutagens can actually increase the rate of mutation. And 2 examples we see, one would be radiation. And this occurs if there's sufficient energy to change the chemical structure within the DNA. Gamma rays, X-rays, alpha particles such as from radon and shortwave, ultraviolet can also, those can all cause mutations within the DNA. We can also see chemical agents be a mutagen. An example being polycyclic aromatic hydrocarbons found in tobacco smoke. Mustard gas would be another example. So chemicals can also cause mutations to DNA and lead to a whole host of different consequences. So as I've said a couple of times now, mutations are random. This is a really key important part, and it's actually very beneficial for the survival of the species and creating genetic diversity. Organisms, individuals themselves cannot choose to or elect to have mutations. They're random. And so individual organisms or a species can't change their DNA sequences. Mutations can occur anywhere within the base sequence, but some regions have a higher probability of experiencing mutations than others because some are more prone to chemical changes, or chemical change like the mutagens that we just discussed, which cause a mutation. Due to randomness, most mutations are unlikely to be beneficial. Usually we think of mutations as being bad. Yes, that can be the case, but generally they're neutral. They have no effect, or sometimes they can be harmful, and on rare occasions they actually can be beneficial. It's not possible to test the effectiveness of mutations that occur within an organism over their lifetime, because only those that are beneficial actually get passed on from one generation to the next. Harmful mutations are generally going to cause the individual to not survive. Traits that occur in sex cells, so genes within sex cells that experience mutations that are beneficial, that doesn't result in the organism dying, will be passed down to offspring. And so we can see these develop and maybe expand within the population over time. So speaking of sex or reproductive cells, where the mutation takes place actually makes a big difference in terms of what type of cell. A somatic cell is a body cell, and this would be a cell that the genes are not passed on to the offspring. So like a skin cell or a heart cell, for example. The mutations that take place within these cells are eliminated when that individual dies. They can still have implications for the individual, as the mutations to the cell cycle controlling genes, proto-oncogenes, can potentially lead to cancer or have other impacts, other effects. So mutations that take place within the body cells, somatic cells, can affect that one individual organism. The opposite of that are the germ cells or the sex cells, and these are cells that give rise to the gametes, sperm, and egg, and they are passed from parent to offspring. So mutations that take place in these cells can have some impacts on not only the future generations, but potentially multiple generations down the road. The new allele from that mutation can be inherited, whether it's positive or negative. So it's really important to ensure that the gamete-producing cells do not experience mutations, particularly in humans. You can think of this as if you have an X-ray, usually you'll have some sort of vest put over reproductive areas of your body in order to ensure that those X-rays are not causing any mutations to that particular region within those cells. While mutations oftentimes get a bad rap, they're actually really essential to the survival of species because they can create genetic variation, genetic variety. And so to understand this further, we need to define the term allele. I've used it a little bit already throughout this video, but an allele is a particular form or type of a trait that differs by one or more bases from other alleles of the same gene. So it's the same gene, it's the production of the same protein, it's just a slight alteration that changes that particular protein. And so in the pictures here, we can see as an example, different alleles are responsible for producing different visible characteristics that we see in humans. Hair color would be a good example, eye color, skin color. All of these traits within humans are coded for or are produced from multiple genes. They're called polygenic traits, but still in the same, each of the differences that we see in terms of hair color are caused by different alleles. And so mutations can change alleles by alternating the base sequence of nucleotides and thereby increasing the number of different alleles within the population. And so mutations are a source of new genetic variation. They are what produce new genetic variation. Meiosis and sexual reproduction also increase the variation by creating new combinations of alleles through crossing over and independent assortment, but these are not mutations. Mutations are what are producing new sources of genetic variation, and so they are essential to species, as genetic variation is required for natural selection to be able to function, and species cannot evolve and change if natural selection is not taking place, and so there must be differences within the population. There must be variation. If the species doesn't have variation, it'll eventually become more and more similar, leading to extinction. As it does not, the species does not have the ability to be able to evolve because it doesn't have any variation. And so mutations can be very beneficial. They can have no effect, or they can have a harmful effect, but they are necessary for the survival of species. And it's important to remember that all mutations are random. As we now turn our attention to discussing genetic engineering and our human ability to change genes, it's important to understand a process called gene knockout. The sequence of DNA nucleotides that encode for a protein are transcribed into RNA, specifically messenger RNA, and they begin, as I mentioned previously, with a codon, which is 3 nucleotides on that messenger RNA, and that first initial codon that starts the process is called a start codon, and it's always AUG. Methionine is the amino acid, and it continues for X amount of nucleotides, and it ends with a stop codon. And these portions that are actually used to be able to produce that polypeptide, that protein, are called open reading frames. Oftentimes we don't know or understand the function of these sequences initially. We don't know exactly what they do. And so using a process called gene knockout, we can use this technique to be able to determine the function of a base sequence by knocking off or turning off the production of that protein. And it's generally conducted in mice. And how this process works is the DNA-based sequence is inserted into the genome of an embryonic mouse as a replacement for the target gene. So thereby it deletes the original sequence. The successful embryo cells are selected, grown into an adult, and those adults have only one copy of the target gene. Males and females with one copy are mated so that 25% of their offspring will have no copies of the target gene, so it's thus been knocked out, and those are referred to as knockout mice. The phenotype of the knockout mice then can be investigated to identify which traits have changed, because that protein, that sequence is no longer within their DNA. That would be the genotype. The genes have been changed. We can then observe the phenotype to see what effect that particular sequence of DNA, that codon within the messenger RNA, what effect did it have? And so then that indicates the function of the gene that has been knocked out. This is important because it can help indicate and identify the function of these open reading frame sequences. The development of CRISPR has really changed our ability to make genetic modifications. And the CRISPR-Cas9 system, you probably have heard of, is a natural system within prokaryotes that's used for finding and alternating specific sections of the genome. And this is primarily within prokaryotes a defense mechanism against viruses. In 2020, the Nobel Prize in Chemistry was awarded to Jennifer Doudna and Emmanuel Charpentier for their work in discovering this system and being able to use it. And now it's widely adopted throughout multiple different scientific fields, primarily biology. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The repeats being the base sequences occurring several times. Short in that they're a short number of bases, usually about 23 to 47 bases. They're clustered. The repeats are grouped together in one part of the genome, and they're regularly interspersed. The repeats are separated by other base sequences of similar length, where each spacer is unique, and they're palindromic, meaning that each sequence has parts which can be read the same backwards and forwards. And so that's the CRISPR portion. Cas9 is an enzyme that can identify specific DNA-based sequences by using a guide RNA to bound to it to be able to read it. And this guide RNA is made by transcribing one spacer and one repeat from CRISPR that is complementary to the target DNA. Cas9 then moves along the DNA by uncoiling it to be able to find its target sequence. And the identification of that target sequence then initiates endonucleases that cut one sugar phosphate bond within the DNA strand, and it allows prokaryotes to then target, recognize, and destroy the foreign DNA, again, generally from viruses that are trying to infect those prokaryotes. In order to use CRISPR-Cas9 for gene editing, there's a number of steps that are required in order to prepare for target sequence identification and modification. And the first portion of that is the preparation of the guide RNA, sometimes referred to as PEG RNA. The 5' end has a usual guide sequence, and 2 extra sequences are added immediately adjacent to each other at the 3' end, one being a primer binding site and an RNA copy of the desired replacement base sequence for gene editing, and this is called the RT template. The Cas9 molecule is made with modifications. Reverse transcriptase is an enzyme that is attached that can assemble a strand of DNA nucleotides that are complementary to the base sequence of the RT template, and then endonuclease in Cas9 is inactivated, so it cuts only one DNA strand, not both of the strands. And so then the use of the Cas9 and the template RNA, the guide RNA, can be introduced into a cell for gene editing. And how this works, the Cas9 moves along the DNA searching for the target sequence by using the 5' end. When the target is identified, a cut is made in the complementary sequence on the other strand to be able to create 3' and 5' ends. The 2 DNA strands are separated and the guide RNA that's linked to the target sequence by base pairing. The primer binding site of the PEG RNA, that guide RNA, binds by base pairing to the other DNA strand on the 3' end, thereby allowing reverse transcriptase to be initiated. That reverse transcription then adds DNA nucleotides from the 3' end in the usual 5' to 3', because that's the direction that DNA is always replicated. And it does that by using the RT template. And then this is the new genetic information that will replace the removed sequence. The 2 strands of DNA then pair up again, and reverse transcriptase displaces the original strand, and it's now just a single flap of nucleotides. Those are removed by editing out the original base sequence, so they're removed from the original base sequence, and then the mispairing of bases where DNA was removed will be reconnected, corrected by repair DNA enzymes to create 2 strands, one with the replacement sequence and one with the complementary sequence. And so then that is the DNA that's edited and modified. This ability has a lot of ethical implications. As of August in 2022, there's no internationally agreed upon regulatory framework for gene editing. Most countries are grappling and struggling with whether to allow it or not allow it, and it really differs by different locations because the gene editing is different from traditional genetic engineering. The largest debates and ethical implications focus upon human germline editing occurring at the embryonic stage. For example, in Japan, this is permitted for research purposes, but it's prohibited in Canada. So it really depends on the country. Generally, it's been accepted that the editing of human embryos is not acceptable. In 2018, a Chinese scientist actually did genetically modify human embryos in secret using CRISPR, 2 twin girls, and the genetic modification was to remove factors involving HIV positivity, and so removing the potential that these 2 individuals would develop HIV. Did this in secret, and the twins were born and are now supposedly healthy and surviving. But the scientist, he was recently released in March of 2024. And so if you want to think of this as kind of like a Pandora's box, the potential for this exists. And now what do we do, and how do we make these decisions that have really big ethical implications as we move forward and this technology only continues to advance? How do we decide what's acceptable and not acceptable within each of our individual countries and also worldwide? To finish our video, we want to look at gene sequences that are similar between different species, and we call these conserved gene sequences, in which they're identical or nearly identical across a species or a group of species. Highly conserved sequences are identical or similar over really long periods of time of evolution, and this occurs over a wide range of species. So looking at different species, we see the same sequences of DNA between those different species. It's typically found in protein coding components of the genome and sequences that are used to make ribosomal or transfer RNA or that regulate gene expression. When speciation occurs, when one species becomes two different species, each of those species maintain these genes and the gene regulators, so the sequences are conserved. And this explains how some sequences are found in all mammal or even all vertebrate species, because they are essential to the survival and they're conserved as species develop and become different species. Non-coding regions of sequences can also be conserved. The conservation of these sequences suggests that they do have some function, unknown at this point, or there's an alternative hypothesis that based on mutation rates not being identical throughout the genome, in which the conserved non-coding elements might be in regions where mutation rates are really, really low. Which of these two possibilities, scientists are not entirely sure. A good example of this can be found in the HACNS1 gene, which has 546 base pairs, and it is highly conserved across birds and mammals, and so it would suggest that it has been unchanged for millions of years because it's very, very similar between all of these different species. And so the ability to edit genes and control the genome of not just other species, but also humans, provides all kinds of really wild and interesting questions that will need to be grappled with as this technology advances in the future, but is really a fascinating aspect of biology and bioengineering and biotechnology.